Okay, and we're here for our Microbiology Journal Club for our October edition. And I am going to apologize ahead of time if I start coughing into the microphone. I'm fighting a cold, and so I hopefully will not, unfortunately I can't make anybody sick because I can't transmit my pathogen through the internet yet. But I will try very hard not to cough into the microphone. So we're here today to talk about, this is our monthly online journal club, and today we're going to be talking about this paper, which is called Spontaneous Mutation Rate is a plastic trait associated with population density across domains of life. So this is a paper in PLoS Biology. It was actually sent to me by a colleague of mine in the predatory bacteria world, Andrew Levering. And he sent it my way because I've been talking about an experimental evolution project that we're doing with predatory bacteria. And he thought that this might be something interesting for us to look at because of the fact that it talks about how mutation rate is associated with the population density. So if you're thinking about setting up an evolution experiment, you are definitely thinking about mutation rates, and then you might want to think about manipulating population density as a factor that influences the rate at which you're getting mutations. And I thought that that was an excellent suggestion on his part, and I thought perfect paper for our Microbiology Journal Club. And it gets us into a little bit of molecular evolution stuff, which I think is good too. So before we get going, I'm just going to go ahead and introduce myself. My name is Laura Williams. I'm an assistant professor at Providence College in Providence, Rhode Island. And Karen is with us, but she's going to lurk a little bit because she might have a student coming by to talk to her. So she might jump in, she might not, but she is there in the little panels. But we also have Nicole with us. So Nicole, do you want to introduce yourself?
Certainly. I'm Nicole Suktio, and I am a postdoc for 5 more days at the University of Northern British Columbia, where I've been working on microbiome methods to sort of look at how forest soil bacteria and fungi respond to very mild disturbances in the form of shallowly dug holes. So some of that's still going on, but I think I'm going to be exploring teaching in the next little while, and it will be teaching microbiology, where I can think about ideas such as those in this paper about what is really driving changes in mutation. And the cool thing about this particular article that we're looking at, and it starts to hint at ideas with metabolic pools that influence things and conserved mechanisms of how mutation is generated according to population density, because they do look at a wide range of organisms. So I am totally pumped to sort of chat about this, even though I don't know if I understand everything.
Excellent, that's great. Wow, 5 days, that's it?
Yes, it is 5 days, according to the contract letter, unless something surprising happens, but I don't think anything surprising is going to happen. So,
oh my gosh.
Yeah, it's an interesting wrap-up time. So
yeah, those transitional times are always challenging in many ways, but hopefully yours goes well.
Mm-hmm.
All right, so what we're going to do is we're going to, as we normally do in our journal club, we're going to kind of break down this paper. And so what these authors wanted to do, and they don't spend a lot of time in their introduction kind of getting into all of this, but what they're talking about is the mutation rate in its own right is a really important feature of evolution, an important feature of population genetics. I thought it was interesting because I feel like you could probably make a couple of definitions of the mutation rate. The way they present it is essentially, I think this is a well-accepted definition of the mutation rate, but it is, it's not only the rate of errors, but it's also taking into account the repair mechanisms. So if you get errors introduced during replication, or if you get a mutation as a result of DNA damage of some kind, my assumption is they're accounting for both intrinsic and extrinsic factors that cause a change in the DNA sequence, but they're also wrapping into this mutation rate, they're also wrapping into that the repair mechanisms that the cell uses. So my read on this is what they're looking at is mutations, changes in the DNA sequence, changes in the bases that remain after the repair mechanisms have had an opportunity to repair it, but they've gotten past those repair mechanisms, and now they are being replicated, they're being inherited. So if you have a population of bacterial cells, you're going to find that some of those cells are going to have that mutation because they divided from the original cell that experienced the mutation. That was my read on it. Nicole, did that match kind of?
Yeah, I totally agree that I guess I hadn't really thought about the idea of where the paper goes in terms of considering mechanisms as to be something that's rolled into their definition of mutation rate, but the consideration mechanistically that what we're actually looking at are genomic changes that happen during replication that stick after the replication event is totally where this is going. And I completely agree with you that what they are operationally defining as mutation rate definitely rolls all of those mechanistic considerations for things that are either evading selectivity or attempting to and failing are also parts of that.
Because I think you could ask the question like, what's the error rate? You know, which is a separate question. It's like, what's the different pieces of mutation? You could say, well, what's the rate at which a polymerase actually introduces a mistake? And then you could ask questions like, well, what's the rate at which this external, UV radiation is going to cause a change? But that's a different, and probably harder to measure, but that's a different thing than saying, all right, after whatever process caused the change, then you have a possibility to correct the change. And now what we're saying moving forward is we're going to measure the rate at which stuff changed and didn't get fixed.
And of course, it's still all being anchored, especially with the fluctuation test part of the paper, by the phenotype. I mean, we can go after it with genomic sequencing, which I do believe is part of this experimental setup as well. But I mean, the initial data sets that were used are based on observable changes in what said organism is doing in culture.
Yeah, right, exactly. And so that gets back to using the phenotype as a screen to try to assess mutation rate. Okay, so I wanted to go into that because I think that's a whole thing you could chew on about, like, what do you mean by mutation rate? But for their definition, I think it makes sense where they're going with this. So then they talk about, as Nicole mentioned, they talk about the fluctuation test. And this is going to be one of the things that they're using in bacteria in particular. They're going to be using as a way of determining the mutation rate. So what they're looking at is the fluctuation test is going to be a phenotypic marker of mutation. And then what they've got here is essentially spontaneous mutation rates have long been estimated in microbes using counts of cells gaining a phenotypic marker of mutation in environments lacking selection for that marker. In this case, what they eventually get into is looking at the development of resistance to antibiotics. So we have a population of bacterial cells. They're not growing in the presence of that antibiotic, so there's no active selection going on. But then what you do is you can take that population of cells, you can plate them on an antibiotic-containing medium, and you can look to see how many of the cells mutated in a way in the gene that impacts whether or not bacterium is resistant to this antibiotic. So there's things like, I'm blanking on it right now because my brain is kind of fogged with cold medicine. I think it's like nalidixic acid. There's a couple of antibiotics where you can get a single base change in a gene, like in the RPOS gene, for instance. And that single base change is going to give you, as a bacterium, resistance to an antibiotic. So this is a way for them to screen for the mutation rate, the underlying mutation rate, because they can do a lot of back calculations about the likelihood of that change having happened in that part of the gene, how big the gene is compared to the genome, and so forth. So there's a bit of extrapolation that has to happen, but I think that's a well-accepted test for trying to get at the underlying mutation rate. Nicole, do you have any?
I was just going to thank you for clarifying like the types of mutations and the types of resistance there, because I mean, it was one of those things where I'm just like, well, there's many different ways this can happen. I probably should have read a little more closely on that, but understanding that we're talking about simple genomic alterations that can lead to measurable types of antibiotic resistance is helpful here, definitely.
Yeah, and that's a good point, because it's certainly true. I mean, antibiotic resistance is a big phenotypically, there's as an umbrella term for a lot of different mechanisms. And this is certainly not talking about things like acquisition of an efflux pump or something like that. These are changes that, and I wish it's escaping me right now, but I wish I could remember the exact
I
one.
actually have, I hate saying this, but it's sort of like an interesting way for me to put it that way. It's just like cancer is the genomic problem we all wished we had, because there's that direct translation of mutation into phenotype, and we're looking at antibiotic resistance mutations that basically emulate that. It's a single base change. It leads to a simple consequence where there's not like a big web of causation around what's actually causing what's there.
Right, exactly.
Yeah.
Yeah, exactly.
Yeah.
So in these cases, and the ones that they used too, when they did their own tests, when they did fluctuation tests with bacteria, for instance, they talk about the fact that they're going to do this with rifampicin and nalidixic acid. And so there's those antibiotics, there are mechanisms by which the target gene, it's not, the gene isn't targeted, it's really the protein product and the pathway that's targeted. But the gene responsible for making proteins involved in the pathway that's targeted by the antibiotic, there can be single base changes in those genes that then lead to resistance to the antibiotic. And so that's how they can kind of measure the mutation rate. All right. This is like, I was thinking about it when I read this, and I was like, this is like the reverse of the Ames test. Is that it? Because like the Ames test is basically, can you get a revertant that goes back? Oh, hey, it's Sagita. Hello. She's getting set up. So let's see. All right, so what we want to do now is we want to kind of talk about how they're setting up their test here. So they said that, actually, let me see if I can, really quick. Whoops. Sangeeta, are you with us? Okay.
Yeah.
All right, so what's that?
I'm in a very noisy environment, so I'm just going to mute myself unless I have something to say here.
Perfect, thank you, yeah. Okay, so so what we're going to do now is we're going to talk about the, they discussed that the density associated mutation rate plasticity. So they've got this great acronym that they've come up with that I'm sure they're very happy with. So it's called DAMP, density associated mutation rate plasticity. And so this is not a new thing that they're introducing in this paper. It's been a phenomenon that's observed before. But in this paper, they're making an effort to test some questions, some open questions about this. So they're going to look at whether or not this is a phenomenon that occurs across different domains of life. And so what they're looking at here is they're going to be using microbes. They do mention that they're not getting into more complex eukaryotic organisms because that's a little more challenging to do. Generation times cause problems. You know, if they're going to do this in fruit flies, good luck. But they're basically going to do this in microbes. So they're going to look across the bacteria, archaea, fungi, yeasts, and they're also looking at viruses. So they just want to see, is this a phenomenon that you can observe in multiple different microbes from multiple different domains, and then the viruses as well, because they sit outside the tree of life. And then they also wanted to look to see, okay, does this vary? If we see this as a trend across domains of life, what happens within something like E. coli? Do you get variation in whether or not this phenomenon is observed? And then the other thing they wanted to do was try to come up with a mechanistic explanation. So if they do find this phenomenon, that mutation rate is associated with population density, what's causing it? Is there a particular mechanism that they can start to pinpoint and narrow in on what is actually underlying this phenomenon? So to answer their first question, they looked at a number of different, they kind of did a big literature search, and they looked at a number of different papers that had measurements of mutation rate and had measurements of the final population density. Now that was my understanding of it, is that they were looking for the final population density in the experiments, and that was what they used as their measurement of density. If that doesn't sound like what other people read it as, definitely stop me. Okay, I think everybody's good with that. All right, so what they did was they actually pulled all of that information out of the literature, and then they came up with this very interesting figure, which I'm going to try and attempt to do a screen share of. Here we go. All right, can you guys see that? Okay, perfect. So what they are showing is basically they've got this figure that compiled data from multiple studies, and they're showing on the x-axis they've got their estimation of the final population density. So it's units per milliliter. If you've got viruses, I think you're probably talking about plaque-forming units. If you've got bacteria, I think you're probably talking about colony-forming units. And then along the y-axis, you've got mutation rate, and then it's per generation. So I had a question before we actually started broadcasting. I was like, but it's mutation rate per what per generation? Like, they don't say. And Karen actually spotted it in the materials and methods. So on the y-axis, it looks like it's mutation rate, and what they say is mutation rate per cell per generation, which I got a little feisty about because viruses technically aren't cells, but that's okay. So we're going to assume that for viruses, it must be mutation rate per particle per generation. Or if you're bacteria or yeast, you're talking about mutation rate per cell per generation. My guess is that this can roughly translate to mutation rate per genome per generation, unless you've got something that's a polyploid. I don't know, do you guys have thoughts on that? Do you think that that's kind of an accurate conclusion?
I think going back to the virus thing that you had an issue with, like, that's a case where we don't know, right? I mean, it's depending on the metabolite pools, which I mean is hinting ahead at some things that are kind of an issue for interpretation of wherever that virus got put together inside, you know? So it's difficult to know sort of what the precision of this estimate of mutation rate really means. I have a question also about that. So with the genes that are conventionally used for the types of tests that are sort of compiled in this particular figure, are they always screening for something that was a single gene copy? Because I mean, some of these pieces of data, I assume, also predate being able to sequence all the things.
Oh yeah, that's a really, I don't know. That's a really good point. What they, like, for instance, what were the target for the viruses? How are they assessing mutation rate in different genes in the viruses? Yeah. I'm not sure.
Well, I mean, also for the bacteria, I'm just going to assume that maybe for some of these it's not an issue because there would be only one, but like, I don't know.
Yeah.
I mean,
but
for some of this.
But if you're looking at like ribosomal RNA, which, you know, probably wouldn't be a good example because it's
absolutely not.
You know, but it has 7 copies. So in some, like E. coli, for instance. So yeah, I don't know. That's interesting. They might have been able to control for it because, to be honest, I did not go through. They have very extensive supporting information, which I admit that I did not go through. So it could be that they addressed that in their supporting information. So yes, authors of the paper, I apologize if we are ignorant of something that you clearly stated in the supporting information. Yeah, that's a good point to keep in mind. I think you made a good point about the precision in terms of I think they concluded that a lot of what they're seeing would probably be robust to some of those factors, but that is a good thing to keep in mind. Other thoughts on that? Okay. All right. So what the whole purpose of Figure 1 is to actually address their first question. Do we see population density dependent variation in mutation rate across representatives from different domains of life, microbial representatives? And I mean, if you look at it, yeah, you know, that's you definitely see they've got a nice slope going. I started to kind of deconstruct it based on the type of microbe, and then the domain. So for instance, you got a lot of your viruses, which are the diamond shapes. You got a lot of your viruses up in the top left corner. So to me, it looks like that's where all the viruses are. So all of the data that they were pulling from the literature on viruses, mutation rate, population density, they're getting low population densities for the viruses and high mutation rates. And that definitely sets the viruses apart from the other microbes. And I kind of looked at those population densities and thought, admittedly, I don't know about when you're doing these experiments with these viruses, but those population densities seemed kind of low to me. I don't know though, but I'm not a virologist, so I'm not sure. I didn't understand why the viruses were so sequestered in the lower part of the x-axis in terms of we only have representatives that the experiment got to a population density of 10 to the 4 and 10 to the 6.
Part of me is wondering, like, whether we're seeing something in terms of what it's representing a range where you can robustly estimate viral particle density. I don't know, but like that could be a thing. I mean, it's still really interesting though to think about this because, I mean, they're in a cell that's making their genomes for them. So like,
yeah,
thinking about what the cell is reading when it's doing all this, and like, is there just a more relaxed filter? Because on some level, it's just like, I don't like. There isn't really a clear physiological, consequence yet until this thing is built up and does things.
Yeah, yeah.
Right, because it's not their own genome. So it's interesting to think about that in terms of like regulatory feedbacks on what those genomes are and aren't doing, because they aren't really playing a part in the host physiology at the time more copies of them are really being made, right?
Yeah, yeah. And I guess it might depend on how these experiments are set up in terms of when did they just, because this is just one, each of these points is just one point, which I assume is the end point of the experiment, and they just took the population density at that point. So that could make an impact on it too. The other thing I noticed about this was we've got a lot of our yeast. So there's only one representative of the eukaryotes, which is everybody's favorite Saccharomyces cerevisiae, your baker's yeast and your brewer's yeast. And it's kind of clustered, all these red squares, they're kind of clustered right at about 10 to the 8th population density. And there's some that kind of go out from that. But I was kind of wondering, like, if you just pull out those samples of yeast, how strong that relationship still is. I didn't see anything in the paper where they pulled out individual, and maybe I just missed it, but I didn't see anything in the paper where they just pulled out individual groups on their own. So like if we just pull out yeast, what do we see? Did I miss that? Did they actually look at that? If they just, rather than having everybody on figure 1, if they just looked at yeast? I think I saw it, but yeah, I thought it was interesting because it looks like, at least in this case, it looks like from the literature, the experiments on yeast didn't show quite as clear of a relationship. It just looks like there's a lot of clustering where the final population density was 10 to the 8th. And then the mutation rate hovers really close. I mean, it's within a couple orders of magnitude, maybe.
This is maybe me getting a bit ahead, but when they did the linear mixed model stuff, was organism as a term one way? They just kind of looked at the overall trend to sort of say, look, this is just how it is for the range of things we've looked at. Like, this is the overall trend. Is that part of?
I think it might be.
Yeah.
I think it might be. Yeah. Yeah. So maybe we'll keep that on hold, and when we get into, because they did their own tests with yeast. So maybe what we can do is when we get into looking at yeast, we can kind of think about what implications that has. But I will say, overall, if you plot all of this data on here, there's definitely a relationship between mutation rate and population density. So I think their major argument was that, look, we saw this in one organism, and what we wanted to know is, if you look across multiple species from multiple domains, and viruses. Do you see this trend? And it looks like you do. So there is a real phenomenon that mutation rate is varying with population density. They're correlated with each other. And I wouldn't argue with that. I think that they're showing that overall. Other thoughts on figure 1? Okay, so let me see. Stop. There we go. All right. So so then what they wanted to do, so they said, oh, hey, awesome. So it does look like, in fact, that we've got this trend. We're seeing it across different domains. So what we want to do now is we want to look at, and let's see. So they said there's a clear negative association between mutation rate and population density. This association, and then this is back to what Nicole was saying, this association potentially involves both between and within organism variation in mutation rate. Okay, so what I interpreted that to mean is between organism is, I think that they were using organism as species. So I'm thinking that what they were saying is between species, this is going to vary, but then within a species, this is going to vary. So my interpretation of what they meant by within organism was actually strain level variation, especially if you're thinking about bacteria and yeast. I fully admit that I'm much less experienced with virology, so I don't know if those are the correct terms to use for viruses, which might be why they were using the word organism. But my interpretation was that they were saying, okay, we think we've got variation between species, but then we might also have variation within a species at the level of strains. Does that sound right?
Yeah, actually on page 13 of 19, there's this whole paragraph under the heading phylogeny used in analyzing published mutation rates. So they actually kind of talk about when they used last common ancestor for delineating things like Saccharomyces cerevisiae, but then I'm trying to like look at this, but my head's just scrambled a bit. So it looks like they did sort of do some sort of consideration of subspecies for a couple genera, like the Salmonella. And then,
yeah, which would totally make sense.
Yeah, and then it looks like with the viruses, which is the last sentence in that paragraph, is collapsing things into common orders. So it looks like they had to make a couple decisions, but they've tried to disclose them about what the organismal groupings were when they went ahead and used whatever their scheme of phylogenetic relatedness was. But I agree with your interpretation generally that I think they're trying to parse between species for between organism, I guess.
Yeah, I think that was my conclusion of what they were using in terms of organism, is just as a placeholder for species. I want to make sure I'm not leaving Sangeeta out. Do you have any thoughts on it?
No, I agree. I agree with what you're saying. I think, for example, if you look at, I also got the same. My interpretation was the same. And if you look at figure 2, for example, then they do those experiments specifically for yeast, and they do it for different strains. And they have all these variation between different strains of the yeast, which is, I guess, what we're talking about. So, yeah.
Yeah, I agree with you. I think that's a good segue into figure 2 then. So what they're doing is they're then going to, with figure 2, and they start to kind of ask questions about, for figure 1, they present some possible explanations for why you would see that relationship in figure 1, and they do acknowledge that you could have things like technical bias, reporting bias. So pulling things from the literature, there's some biases that could be introduced that would potentially lead to the relationship that we saw in figure 1. And so as a way of trying to actually demonstrate that those biases were not affecting it, they actually did their own testing in the lab with E. coli and then with Saccharomyces cerevisiae, 3 different strains of Saccharomyces. And so they said, well, if we do this in the lab and we can apply our own model consistently across all of these tests and we still see the same phenomenon, then that's a way of rejecting these hypotheses that have to do with just technical bias, reporting bias, so forth. So what they did was they, this gets to the fluctuation test. So they said, using fluctuation tests, we estimated mutation rates in batch cultures at 2 marker loci in each organism. So in E. coli, they did RPOB and the gyrase gene. And then in Saccharomyces, they did the 25S ribosomal proteins and something called URA3. And these genes confer resistance to, in E. coli, it's rifampicin and nalidixic acid. In yeast, it's hygromycin B, and then 5-fluoroorotic acid, because I think that's how you say it. And so what they did was, and I'll pop this up here, they're looking at using those fluctuation tests, so using the acquisition of a mutation that causes antibiotic resistance as a way of measuring the mutation rate. And then they looked at the population density as well to see if they could see the same phenomenon, which, all right, so we've got the bacteria up there at the top. The E. coli is in purple. We haven't talked about the blue yet, so we'll get back to the blue because that's Pseudomonas. So the E. coli in purple is basically what we've got is triangles are mutation rates for rifampicin resistant, and then the circles are mutation rates that ended up with nalidixic acid resistance. So based on either rifampicin resistance as the triangle or nalidixic acid as the circles. And what they're seeing there is essentially what we've got is this test of significance that they do is essentially to look at what's the probability that the slope of the line is 0. So their null hypothesis in this case is the slope of the line is 0. You know, can we actually reject that with the data that we've got given the slope that we're seeing? And so for E. coli, let's see. So they found for E. coli that the p-value that they got for E. coli is just, you know, ridiculously low. So they felt, and you could see that from the figure, they feel pretty comfortable saying, yes, this data actually supports the idea that the mutation rate is varying with the population density. So for E. coli, it looks like, yeah, we're actually seeing this. We're varying these conditions. We're getting these different final population densities. And the mutation rate, as we're measuring it with the fluctuation test, seems to be varying with the population density. And then they get down and they do this with yeast. Haha, I just tried to scroll the thing on my actual video rather than the other thing. I confused myself. So with yeast, they've got these different strains of Saccharomyces. And so they've got 3 strains. The squares are going to be mutation rates to get hygromycin resistance. The diamonds are going to be mutation rate to get the FOA resistance. And then they've got brown, orange, and red in terms of the different strains of Saccharomyces. So one of the things they noted here was like, well, that's interesting. It looks like, so I think they found, and I thought I had a note of this. I think they found that 2 of the strains have a pretty significant, I mean, you can see this from the figure, 2 of the strains have a pretty significant slope, but then your strain in brown, I can't remember if that one was actually significantly different from 0 or not, but you can definitely tell that there's some variation. Let's see. So they're calling this, they're saying that basically what's happening is within Saccharomyces cerevisiae, you're seeing plasticity in this phenotype. You're seeing that some strains demonstrate density-dependent mutation rate variation more strongly than other strains. Thoughts on this?
I'm trying to look at it right now to see if there's within strain differences depending on the type of resistance they were selecting for, and like,
oh,
I could be reading this wrong though, because for the brown represents, that's
yeah, you're
just one
right.
strain, yeah.
Yeah, you're right, because they put those together. Yeah, interesting. So they don't have trend lines for, they've just got trend lines for the strains. They don't have trend lines for the strains
Yeah,
plus
because
the
I don't
antibiotic
see
resistance.
brown diamonds, and I'm wondering if I'm just missing something obvious about them.
No, I don't think
Not,
so.
no.
Hmm.
Because the thing I find sort of interesting about considering that is that I just did a little bit of Googling. So this 5-fluoroerotic acid does get converted if you are sensitive to 5-fluorouracil, which is a pyrimidine antimetabolite. And it's actually like, it's an anti-cancer drug. So like the idea with that is it's supposed to interfere with replication directly. So to me, it kind of intuitively makes sense that like,
Oh,
there could be
interesting.
some interesting interplay of like the types of mutation rates you're going to get with that just because of what it is chemically and what it's doing.
Yeah, definitely.
Even though the thing that's really interesting to me is when you look at the orange strain, I mean, from the little I can sort of see there, there's a lot that's obscured by the light orange or the light yellow boxes, but for that one that's sort of the more blood orange color at the bottom, it doesn't
Yeah.
actually look like that's a difference. Like it looks like you kind of are getting things that are averaging out to the same slope, although it's really hard to say. I mean, I'm eyeballing data and saying very bad things about it non-analytically, but it's kind of interesting to think about that.
That's what we're here
For me.
for. We're here to eyeball data and make broad conclusions that are not
Yeah.
statistically supported.
Yeah.
But no, but you're right. I mean, it looks like, because they did mention too that the open symbols have to do with, you know, there's some bounds. There's a lower bound and an upper bound in terms of their estimates that are traditionally people feel comfortable with.
Okay.
And I think those symbols are bringing in data that sit outside those bounds or just at those bounds. And so they've tried to kind of denote those as being like, you know, we understand that these are data that might not be represented in other people's studies. That was the way I read it, at least.
Yeah.
I'm always afraid. I'm always afraid that during one of these journal clubs, I'm going to be making this broad statement and the author of the paper is going to watch it and be like, oh my God, that's not what we said at all. Why did you say that? So if I've made a mistake, authors, definitely let me know.
Yes.
Yeah, that's interesting. So, I mean, I think you can definitely, their major conclusion from the yeast experiment was that you're getting strain level variation in this phenomenon, which I do agree with them. The strength of the relationships for each strain, I think you could ask some questions about like which data were included and why, you know, the resistance, which resistances were represented or tested for. But I mean, I think what they are showing is that you're getting strain level variation. And they go back basically, and they if we scroll back up to the bacteria, what they decided to do was they decided to test another bacterial genus. So they bring Pseudomonas into it. And so these light kind of teal colored triangles are doing the same fluctuation test, doing the same estimate of mutation rate versus population density with Pseudomonas. And with Pseudomonas, because I remember looking at it, they were basically like, yeah, that's just nothing. So when they do the statistical test to try to reject their null hypothesis that the slope is 0, they can't reject their null hypothesis that the slope is 0 with Pseudomonas. So Pseudomonas, you could argue, doesn't even seem to display this phenomenon at all. So from this, they can basically say, okay, E. coli shows this density-associated mutation rate plasticity. Pseudomonas, at least in the conditions, with the Pseudomonas we used, in the conditions we used, we don't see it. And I think that's an interesting question because you're looking at, now you're trying to figure out what's influencing this. Why are we seeing this? What's the mechanism that's showing, that this phenotype is showing up in some bacteria or some yeast and not others, and that it's showing up more strongly in some strains versus others. That leads into figure 3, but I don't want to move into figure 3 unless we're ready. Does anybody have any final thoughts on figure 2?
Hi, Laura. Can I jump in a little bit there?
Absolutely.
So I was thinking that is there any chance that there are certain metabolite biases between strains? So, but the thing is that I don't actually know anything specifically to Pseudomonas because they're talking about mutation rates. Though only mutation rate they measure in Pseudomonas is to rapamycin, and so they don't actually measure the other ones in Pseudomonas. It's only, so I was wondering, like, is there any chance that within species there might be already a natural predisposition to metabolize certain antibiotics, so different pathways between different species, that that because of their choice of chemical that they decided to use for this test, that it would result in a bias?
Oh, that's a good question. So I am trying to think. So my initial answer would be, from what I understand, I think with these antibiotics that they chose, I think the reason they chose these is because it's kind of a, and I could be very wrong, but I think it's because it's basically a binary. So you can either survive this antibiotic or not because of the mechanism of resistance. So there are a lot of mechanisms of antibiotic resistance, and with these, I think, because for instance with rifampicin, I think it's protein synthesis. So I think it actually binds with part of the. I could be totally wrong. I could be making this up. With rifampicin resistance, I think it actually binds to part of the ribosome and interferes with protein synthesis. And so if you can make a structural change in the ribosome, then the antibiotic can't bind. And so that means that you are then resistant to that antibiotic. I'm just looking that up because if I'm wrong about that, I really want to make sure that I'm not.
I just found something that said changes to the B subunit of RNA polymerase. So I think.
Oh, RNA polymerase. Okay.
Yeah.
Oh, okay. So So it's transcription then, not translation. Is that yeah? So we're okay. All right. So I think it's so it says resistance to rifampicin arises from mutations that alter. I'm on Wikipedia. Residues of the rifampicin binding site on RNA polymerase, resulting in decreased affinity for rifampicin. So that's why they've got the RPO gene targeted here. So they're looking at mutations in the RPO gene, which is the beta subunit of RNA polymerase. So if your RNA polymerase acquires a mutation, then rifampicin can't bind to it, then it can't interfere with transcription, not translation, and then you've got resistance to it. I think the other one, I think nalidixic acid is interferes with DNA gyrase, if I remember right. And so so to get back to your question, Karen, I don't know. I don't know if it's so other things that could be going on is that you could have other mechanisms of resistance. You could have differences between E. coli and Pseudomonas in terms of how well rifampicin gets into the cells. So I suppose those are all questions you would have to think about in terms of, is this fluctuation test actually giving you just the mutation rate, or are there other factors that are potentially influencing the final population density because of the action of the antibiotic, how well the antibiotic gets into the cell, whether or not there are other mechanisms of resistance that could be coming into play. I do think you're right about that point. That was really long-winded. Did that help?
That's right. No, I think when you said the different mechanisms that the antibiotics get into the cell, I think that was probably the key, because we have no, I'm not sure of, this is because I don't actually know what, in terms of Pseudomonas, like Pseudomonas is gram negative, but I'm not sure if it has, I don't know if it has I don't know if it has a capsule, or if it has any other, if it has specific things that would prevent or the antibiotic from coming in, or could have mechanisms to pump the antibiotic out. I'm not actually sure.
Yeah, and I think those are all questions that could lead to interference with the test. So if you're doing this fluctuation test, you want to make sure that you've tried to control for some of those variables, so your data is not getting influenced by all those other phenotypes that you're not interested in measuring. I think that is important. Okay. All right. So what I think I'll do is, so I think this was a neat part of the paper is they said, and this is always the big question, is like, what's the mechanism? Like, what's the biological mechanism for this phenomenon? And they actually went about trying to piece together what is the biological mechanism. And so they started in E. coli, they started making knockouts in DNA repair systems. So they would just disrupt a gene in a DNA repair system and say, does this interfere with DAMP, density associated mutation rate plasticity? Does this interfere with DAMP? Does this interfere with DAMP? And I think what they're trying to look at, so they decided to take the angle of, if you're thinking about mutation rate, the way they've defined it as errors that get introduced plus the organism's ability to correct those errors, that's the mutation rate. So we're looking at the mutations that make it through both those phases and are still found in cells that divide from that population. What they decided to look at was the piece of it that's basically repair. So can we figure out if repair mechanisms are involved with this DAMP phenotype? And so I'm going to pop this up really quick. So they did, they looked at a number of these different pathways in E. coli. And what they found, which I thought was pretty cool, is they knocked out a bunch of stuff that it was like, nope, nope, everything's still DAMP. Which is great, you get to say that. Everything's still DAMP when we knock this stuff out. With the exception of, they've got these light blue circles and the dark blue circles, which are proteins involved in methyl-directed DNA mismatch repair. So those, so if I understood this correct, definitely stop me if I didn't. My understanding of this is if you knock out parts of the methyl-directed mismatch repair pathway, or genes that are in protein products that are important for that particular repair mechanism, you abolish the DAMP phenotype. And that is shown by the fact that the slope of the dark blue and light blue circles is approaching 0, instead of having this very clear slope that shows the relationship between mutation rate, population density. So now that that slope is approaching 0 in your MMR mutants, they're suggesting, we think that that means that this particular repair mechanism is tied to the production of this phenotype. So this density-associated mutation rate plasticity seems to be related to having a functional methyl-directed mismatch repair system. Does that sound right?
That was the interpretation that I got from that figure, and from their comparison of ways that DNA can get messed up that they tested for by their deletion mutations.
Okay.
Yep.
All right. And because we're coming up on 4 o'clock, so then I wanted to just go to figure 4. Sorry if my scrolling makes anybody ill. Because then they said, okay, well, what's going on with that? And I have to admit that I did not have a chance to do as much review of methyl-directed mismatch repair as I would have liked before coming into the journal club this week. But essentially what they're doing is they're now going, all right, so let's see if we can take a look at exactly what's going on. So we would like to know, they're looking at, they kind of split the, I don't know how to say this, the phases, I guess, of this process into 2. So they've got at the top, they're showing mutation avoidance on the left, and they're showing mutation correction on the right. And so what this comes back to is mutation avoidance on the left has to do with, it's basically scavenging this 8-oxo-D-GTP, which is a mutagenic nucleotide. They're scavenging it from the free nucleotide pool. So mutation avoidance means this mutagenic nucleotide is in the free nucleotide pool. We've got a mechanism to actually remove it from the free nucleotide pool so that it doesn't get incorporated into the DNA and doesn't cause problems. And that's mutation avoidance. The other phase of it is if that mutagenic nucleotide, that 8-oxo-DGTP, does get incorporated, then there's a mechanism that's the mutation correction mechanism that tries to fix it once it's got incorporated. So they kind of split this into two phases. Let's get that mutagenic nucleotide out of the free nucleotide pool in the first place, or, okay, it got incorporated into the DNA, so now let's fix it. So then what they did is they did knockouts of, at the top, what we're looking at the top two panels is, I think that's in just E. coli. So we're looking at the genes that are responsible for both those mechanisms. If they knock those genes out, do they still see DAMP? And so what they find is that if they knock out the genes that are associated with removing the mutagenic nucleotide from the free nucleotide pool, that's on the top left, the slope is now pretty much bang on at 0. So the mutation rate is always the same all the time in any population density, does not matter. I mean, that's obviously not true because we've got points going around all over the place, but the trend line is basically showing you we don't really have this population density variation affecting mutation rate variation. But if you knock out the genes that are responsible for correcting, if the mutagenic nucleotide gets incorporated, you still see that DAMP phenotype. So they're arguing that mutation correction, once incorporated, once that mutagenic nucleotide incorporated, is not important. It's actually removing that mutagenic nucleotide from the free nucleotide pool. That's the important part of getting this mutation rate, or population density associated mutation rate variation. That was a lot. That was a lot of talking. Did that make sense?
That totally made sense. And this is actually the part in the paper where I was just like, this is really, really like kind of awesome, because there's so many questions underlying this. Because what does density actually mean then from a metabolic standpoint? It's just, you know, it's getting things out of the pool, not specific enzymatic DNA or DNA replication activity interactions that are dictating how we can fix things. So this idea of like, what was the growth phase at when you've got these higher mutation rates? Which kind of makes sense, because by the time you hit higher densities, you've either got a limited number of the 8-oxo-DGTP, that's one interpretation, or it really just doesn't matter because whatever's happening to pull it out is constrained the whole time. You know, like there's just a point that maybe you can't pull it out fast enough.
Yeah.
I don't know. You know, like, yeah.
I got to the end of this and I was just like, wait, what? Like because they said this too. They're like, this is kind of a counterintuitive phenomenon, right? Mm-hmm. Because you'd think that.
I don't know though, because I mean, there's a lot of hypotheses. And again, I'm going to go back to cancers, thinking about, okay, so when cells are metabolically rejigged, which is one feature in some of the stages of getting to malignancy, which I know is a very eukaryotic paradigm, you're kind of changing the sloppiness of what's there as far as a substrate pool if things are faster. So like, how does this fit into, I mean, they're trying to link it to something that's really, really multifactorial, not straightforward, which is like mutation rate, which is also the collection of mutations, where they're happening and where you can nip it in the bud. So like, it's something really coarse, it turns out. It's just like, what's in this pool of nucleotides and can we pull them out?
Yeah, but I think one of the things, because if you're thinking about bacteria, and you're thinking about the evolution of things like antibiotic resistance, and you're thinking about bacterial population genetics, like one of the things that people talk about is oh God, the name is escaping, but it's like hypermutable strains, like strains that have damage in their DNA repair mechanisms. And that means that they have kind of a higher mutation rate than your average bacteria. And then it's always discussed in terms of the evolutionary advantage. Mm-hmm. You know, what's the trade-off? Like you've got this higher mutation rate so that you might be able to adapt more quickly when you've got these selective pressures. But at the same time, you do have the chance that you're going to be damaging some of your important systems. And so you could get deleterious mutations that can't get removed and da-da-da-da. And so when I was reading this paper, I was kind of approaching it from that perspective.
Yeah.
And I appreciate in the discussion, they have one paragraph that starts, "The evolutionary causes of plasticity and mutation rates need not be adaptive."
No.
People.
No.
Because I'm sure they knew, I mean, that people were going to read this and go, well, what's the evolutionary advantage of blah, blah, blah. And they're saying, you know, nonetheless, mutation is an evolutionary mechanism. So any plastic variation in mutation rates will have consequences for evolutionary trajectories. And I think that's, I have to admit that I did not have a chance to really sit with this paper enough to kind of think about, because I think there's a lot here in terms of broad implications for how you think about this phenomenon, but also how this phenomenon impacts evolutionary trajectories. And I haven't had a chance to kind of sit with it and think about it, because I think they're making important points in the discussion that they're saying this, you know, you may not, it may not be a good idea to spend a lot of time thinking about what's the evolutionary advantage of having damp. I mean, they're saying this you know, ignore that. I mean, I get it,
yeah.
It could be a spandrel. I have no idea. So,
but it's also a big deal, because what you do in a fluctuation test is how a lot of bacteria don't spend their lives in the wider world. So you do actually have to step back and be like, okay, as far as just the mechanism of generating within genome sequence diversity via mutation, I mean, not everything gets to sit in culture for that long. Sometimes you're lucky to get 5 minutes of hydration. So like, are we getting the generation times? Like, what is your net effect size for these sorts of things taking place out in the wider world? And I mean, I come back to soil when I think about that, but there's a lot of compartmentalization. So like, how does this even fit into what everybody wants, which is a predictive scheme, or as they mentioned at the end of the paper, a way of controlling resistance? You know, it's hard to say.
yeah, I completely agree. And so I'm loath to keep people beyond the hour that we agreed upon. But so I'll say for my final thoughts on this, I think I'll reiterate that I hope I have time to sit with this paper again, because I do think what they're getting at here, I want to just be able to absorb it and what its broader implications are. So I will say for me though, because the original reason that this got on my radar is because I mentioned a colleague that sent it to me in relevance to our experimental evolution project. So damp, that phenotype itself and this plasticity and mutation rate may not be adaptive, but if I'm setting up an experimental evolution project in which I am, you know, what we're doing is we're trying to see if you have a predatory bacteria isolate and you give it the same prey over and over and over and over, do you start to see adaptation to that prey? Do you see shifts in things like its efficiency at killing that prey? And to measure that, we're relying on, I mean, mutation is the raw material for selection to act upon. So while this phenomenon and the way they're talking about it, they're like, this may not be adaptive, it's worth it for something. What I'm trying to test is there adaptation to something in my experiment? And so it's worth thinking about how, if this phenomenon is occurring in our bacteria, how the population density is affecting the mutation rate, because that's potentially affecting the rate at which we get this raw material for the selection that we're setting up in our experiment to act on. I'll stop there. Nicole, do you have any final thoughts?
Not many. But I echo your sentiment that I hope I can find time in the next few days, or just keep this paper on my radar to think about, because coming out of the academic system as a biochemist and thinking about, well, we study all this physiology that's canned because we purify these enzymes, and like we don't really know how they're integrated, and then to go and see something like this where, you know, it was really fortuitous on one level, but really insightful to think about just the nucleotide pool, like that key thing that comes into making your genomes, is really where you start to see how a population dynamic, how an aggregate physiological configuration dynamic is affecting what you're seeing generation to generation in terms of what your genome permutation is going to be. It's really a lot to think about in terms of how we need to think about physiologies relating to genomes and genomes relating to what's an organism going to do, you know? So, well, there are some huge pieces there that I think I need to dismantle some of my assumptions around, or like think about what does this mean for experiments? Because all of the densities also were made in the fluctuation tests by manipulating your culture conditions, which is also changing your substrate context, which is also changing your metabolite pool for incidental modifications, such as those that lead to 8-oxo-DGTP. So there's a lot to suss out.
Yeah, definitely. I agree. I don't want to put Karen on the spot, but I did just want to give her an opportunity if there's anything she wanted to say.
No, I'm good. Not in particular. So I think that, yeah, so I totally agree that I definitely need more time to sit with the paper and to absorb what it's actually saying, or the implication, the long-term implication. But yes.
I just accidentally muted myself instead of unmuting myself. Okay, so with that, I think we'll close it out here. We are a monthly journal club, so we've got in the U.S. we have Thanksgiving on the 23rd of November, so we could potentially do our last Thursday of the month again, because that would be November 30th. So I think that's what I'm going to try to do, just to be kind of consistent. So I'm going to aim for November 30th at 3 p.m. Eastern time. For the folks that follow us that are interested in joining, if that's a good time for you, let me know. If that's a bad time for you, let me know. And as always, if there's something on your to-read list that's up at the top that you keep looking at and going, I need to get to that paper, feel free to suggest it, because that's usually how I end up knocking stuff off my to-read list, is I make it a journal club paper, and then I have to read it because I have to talk to you guys about it. So it works out really well. We're always happy for paper suggestions. So with that, we'll close it out. And thanks to everybody for being here. And I really appreciate it. I think this is an interesting conversation about a paper that I'll probably be chewing on as I continue to think about it. So bye, everybody.